This is a quick demonstration of some of the features of the Atlas of Living Australia's spatial portal. What we're looking at now is what's called the data dashboard. It gives us some indications about the nature of the data that's sitting in the atlas, from data sets, record types, collections, records by date, 1629 to the day of recording, species lists, spatial layers. Contextual layers, by the way, are classes like state and territory, whereas environmental layers are continuous values like temperature or solar radiation or rainfall. We do have marine as well as terrestrial. We do not have freshwater yet. We're getting about 50,000 records per week. We're up to about 33 million, and you can look at the breakdown by taxonomic group if you want to. But let's just go straight to the spatial portal and start showing you what we can add to the map. What I'm picking up here are, in fact, all the cobra snake species in Australia, a family called Elapidae. And like everything in the atlas, you are capable of limiting it to an area. And in this case, we'll just simply say world. And here are all the observations of those particular species. You can see some of them are marine and some are terrestrial. A good coverage. Australia is pretty famous for its poisonous snakes. The sort of thing you can do here in what we call the legend is we can look at a density plot or points. You're looking at the points obviously at the moment. We can change the opacity of the points. We can change the size of the points. You can change the colour according to a user-defined setup, or in fact you can break it down as we're going to do now. We'll call faceting by, in this case, the species. So here's the breakdown of the species of those snakes from greatest species count down to the smallest. You can see there are still 133 more species that are not listed with 10,751 records. That just gives you a bit of an indication. You can of course map assemblages just as easy as species, but we just don't have time to do that. So what I'm going to show you now is how you might define the areas. There are 14 different ways of doing it in the atlas. It's very important because we believe that you need a flexible way of defining areas. What we're going to do is just give you two examples. One using a polygon, so we'll make it any shape we like. This is the simple form of that polygon. You can save this or export it if you want to. By default it is called My Area. The sort of thing you can do with it, just to give you a bit of an indication, is to say, well, give me an area report. There you go, 22,996.59 square kilometres, 1,358 species, 15,236 occurrence records, of which we could map them all as a new layer if we wanted to. No expert distributions, no checklist areas or species. 2 documents relate to the species in that area, which we don't have time to look at. So it gives you a bit of an idea. Let's just do something slightly more complicated, but very neat. We can define the area on the basis of environmental layers. In this case, what we might do is look at the seasonality of precipitation. So that varies from 9 to 141. We can change that for the whole of the continental Australia. We might go for 20 to 22 and update the species count. So what it's doing now is looking through the whole of the 33 million occurrence records and also the spatial distribution of that particular layer, given those restrictions, and there is the areas in Australia which conform to seasonality of precipitation between 20 and 22. We can get more complicated if we so desire, and we do desire here. We'll take temperature of the warmest period, and it's telling us here is the range, 13.3 to 42. What we're going to do is to take 25 to 27. Update the species count again. What this is, by the way, are 2 of the characteristics of good Pinot Noir areas of Australia, or the environmental characteristics. So now we're down to 10,153 species in the combination of those restrictions environmentally. If we're happy with that, which we'll do for the moment for the demonstration, we can simply create a new layer which says there is the areas defined by those two characteristics in Australia, and a little sliver on the nullable plain, which suggests they are good areas for growing Pinot Noir. But that's not the full story, that's just two of the characteristics. Okay, so what about looking at the layers themselves? Let's just take a couple of examples. What we can do is look at one terrestrial, distance to permanent water, weighted. What you're seeing here are traffic light indications which show you the relationship between the selected layer and all remaining layers. Green says unrelated, red says highly related, and yellow says there is some relationship. This is fairly significant in a lot of the analytical procedures, and I thought it was quite a neat idea to add it into the spatial portal because it gives you some indications of the relationships between all layers in the system. And there's the layer in question. The only thing we can do with this layer is change the opacity. If you want to look at it a little bit more detail, or if you want the background to come through, you can select any combination. We can zoom to extent, we can look at the metadata of the layer, we can junk the layer. This time we'll simply hide the layer, and I'll show you another example, in this case of a marine layer. Helps to spell it right. This is one of the global marine layers, in fact, and there it is. It shows you what the range of values are in, and again you can just change the transparency as to however you want it. So let's look at the last characteristic here, facet. So this is exactly the same as the legend facet. In this case we're saying let's look at the world. Let's pick a facet type. In this case we'll look at life form. One of the characteristics for the records in the atlas, and this is a breakdown by that life form. Let's just look at the fungi, of which there are 169,000 records roughly, and map that facet class with the life form equal fungi. And it behaves like any other layer in the atlas. We can increase the opacity, we can do further faceting. In this case, let's look at it by collection. So it's going to colour the occurrence records by their collection. Australian National Herbarium has the most, Australian Tropical Herbarium has the least, with 2 records. Okay, let's do some analysis at this stage. We can just switch those layers off and start to look at the tools that work on that data. You've seen an area report. The nearest locality simply takes the nearest 5 points in the gazetteer, of which there are 800,000 in the atlas, and gives you the bearing and direction. Points to grid is an interesting one. It allows you to create sites by species matrix by taking the point occurrences in the atlas and throwing a grid over them, and that grid can be a moving average or a straight grid of the moving averages. You can change the characteristics from a 3x3 to a 9x9 and a series of other characteristics, and also that process will create a species richness layer and an occurrence density layer in addition to the sites by species matrix. Scatter plots, very simple but very powerful. Let's look at the world area. In this case, let's just search for an individual species, and let's just put down one of the sandalwoods. Let's maybe pick Pauciflora as quite a common one, but 195 occurrence records here. We don't want to highlight that. Well, what do you want is the environment. We'll take the 2 simplest ones in the atlas, mean annual temperature and annual precipitation. If you know the shortcut terms, we will display the background environments that are possible in the area defined by those species distributions. We will, in fact, may add another species, another leptomeria, another sandalwood. Squirrelosa. Oop, we have to spell it correctly. There it is there. And we'll just call it my scatter plot. We can rename any of the layers at any time, by the way. So what this is doing is showing you the distribution of the primary species, in this case Leptomeria pauciflora. But the grey background circles are the background species, which are Leptomeria squirrelosa. So it's showing you the relationship between those two species. So you can see there is some overlap, but there's also areas where, in fact, pauciflora occurs, but squirrelosa doesn't. Like anything else in the atlas, we can, in fact, change the characteristics of those points. We can increase the size of the points. We can, in fact, even facet them if we so desire. In this case, we're going to facet them by the bioregion. And so it's colouring up the points accordingly, both in the environmental space and the geographic space. Let's just increase the size of the points on the map. The sort of thing we can do is to say, well, where does this particular occurrence record occur given that in particular environmental characteristic? And there it occurs on the south coast near Albany. So, and we can do the same here. We can actually select areas in the geographic space and see them in the environmental space. The background, by the way, are the potential combinations of those two environmental variables, and the colours of the background, in fact, represent the spatial area. In other words, the lighter colours here represent large geographic areas of those environmental combinations, whereas the black represents small spatial areas of those environmental combinations. We can do a lot more. We can, in fact, subset the records even in environmental space. In other words, we can slice out the in and out layers, quite happily create the subsets. But enough of the scatter plots. Let's do an environmental classification, possibly of just this area will do. So we can, in this case, use current extent, which is one of the definitions of the area, one of the 14. We can, in fact, select what I've pre-identified as 5 rather good independent environmental layers, which cover most of the variation in Australian terrestrial landscapes. You can see the colouring accordingly. We pick the number of groups we want. In this case, we'll just simply do 50 groups, and these are the layers. And what it is actually doing is taking the 5 environmental layers and combining those into one new layer, which represents most of the variation of the 5 layers. Why is that useful in a tool that is aimed at Australia's biodiversity? Well, the simple answer is that we do not have systematic, extensive biodiversity data for the whole of Australia that would allow us, for example, to set up reserves or, in fact, manage existing reserve systems. So what we might want to do is classify the environment and use that classification as a surrogate for biodiversity. So what we're going to see in a second is such a classification of that visible extent of the area of Australia. You can see it a little bit better by increasing the opacity. Okay, so just one other thing I could show you. You can import your own data quite happily. Call it what you will. In this case, we'll simply go to the desktop. There is a file on there. If we sort it, call demo points. There's 100 points. We can restrict that. In this case, those points, by the way, random, and there's a series of different facets on those points. And you'll start to see they are both terrestrial and marine, but they have a series of characteristics in them. In other words, you could include a bunch of other data. For example, if I simply say, well, facet on whatever was in the data set called label 1, which in this case was 1st, 2nd, 3rd, 4th, 5th and 6th, and this shows you in fact the number of occurrences in that breakdown. So there is the simple example demonstration of some of the tools in the Atlas. This is a brief demonstration of the spatial portal of the Atlas of Living Australia. What you can see on the screen now is what we call the data dashboard. It gives you some idea about the nature of the data that's held in the Atlas of Living Australia, how many different data sets, the different types of records, in other words human observations or DNA sequences, preserved specimens, the collection types, and you can drill down through each of these, recording by date. In other words, the first is in 1629 and the last was in fact the day I'm recording this. List of the number of species. Spatial layers, these are both contextual, which really mean they are layers, polygonal layers like classes, or environmental layers like continuous values. An example of the former would be states and territories or bioregion. Example of environmental layer would be mean annual temperature or precipitation. You can see we have a mixture of terrestrial and marine layers. We do not currently have any layers specifically for fresh water, but they're on the way. Breakdown by state and territory, we have a series of identification keys, most recorded species, DNA sequences. We have an Australian version of the Biodiversity Heritage Library, and the breakdown is as such, 38 million pages. The volunteer portal, so these are areas that, projects that have been helped along with volunteers in transcribing records from taxonomic studies. Records by life form, so you can see we have 840,000 fish, for example, as against 209,000 fungi. You can see we're building up quite considerably over time. We tend to get about 50,000 records per week at this stage, so it's an awful lot. And if you want to know more about how many records are in each taxonomic group, you can do the breakdown. Okay, let's just have a look at the spatial portal itself. So up here you've got the standard set of menus, a basic set of map options. This is for the base map. And we'll just have a look through some of the basic characteristics of the spatial portal. If, for example, we might want to look at a particular group of species, you simply need to enter the value into the autocomplete, and in this case it knows that the common name for this family is cobras and snakes, in other words, 43,826 records. So we select that, type next. We could, by the way, create an assemblage of multiple species or even multiple taxa. What extent do we want this mapped over? And the answer is world. And there are all the snake occurrence records for Australia. We can do some very neat things with this. We can change the size of the symbols, we can change the intensity of or the saturation of the colour. We can in fact even look at various components, like show me a breakdown on the basis of cobra snake species, and there's the breakdown by decreasing count of occurrences. And you see the spatial distribution of the different types of species. We can here even display the spatial uncertainty. I'm not going to bother. Or if we want to, we can pick on a particular species, this one here, and just create a new layer of the subset of that particular species. So in other words, if we wanted to just look at that new subset, we can look at it in just that way. So going back, you can see up here we can switch the layers off and on. We can zoom to extent. We can find out metadata about the layer that's being mapped, or we can bin it or just delete the layer. So let's just go back and show you another facet. We can have a look at the collector, so you can see the people who are involved in fact collecting these snake species. My admiration goes out to them. Okay, let's have a look at the second thing. Let's have a look at how you might want to define an area. We have 14 different ways of doing it. Let me just demonstrate 2 of them. You can in fact create a very complex polygon if you so wish, and it creates what's called the well-known text or WKT version of that. So if you want to copy and paste that for future reference, you can do so. You can export it in fact. By default it is called My Area, but you can rename the layers here anything you like. The sort of thing you can do with areas is, for example, to area reports. It understands the last thing you did was the area, but all areas designated are listed. What it's currently doing here is showing you the area in square kilometers, how many species occur in that area, how many occurrences occur in that area. We can map the lot. There are no expert distributions in the area, no checklists, areas, or no species, and it's got 2 documents that relate to that particular area and biodiversity, which we haven't got time to go into. So all defined areas you can do the area report on, or restrict any analysis by those defined areas. Let's take one that's slightly more complicated, but I'm sure you will see the benefit of this complication. Let's simply define an area on the basis of the environment. In this case, I'm going to look at 2 variables, and if you know the shortcut to get to them. Seasonality of precipitation. It'll list here what the upper and lower bounds of that particular layer are, and what we're going to do is just limit it between 20 and 22, update the species count, what it's doing now is running through all 33 million occurrence records and defining the area that corresponds in Australia to the seasonality between 20 and 22. If we want to get more sophisticated, we can add more layer criteria. In this case, temperature of the warmest period. The system will tell us it's between 13.3 and 42.2 centigrade. And what we can do is limit that between 2 temperatures, update the species count again. What this is, by the way, are 2 of the characteristics for the climate, well, environmental conditions for growing good Pinot Noir. So we can see now the area defined by those 2 characteristics alone. We've now got 10,153 species in the area defined by those 2 limits. We can simply say at this stage, just for taking a shortcut, that's enough. Let's just see in Australia where those areas might occur, and there they are, highlighted in red. Okay, so that's at least a very rough indication where it may be quite interesting to grow Pinot Noir. So there's 2 examples of defining area. What we can do is have a look at the layers that are in the atlas now, the environmental layers. So you can do a listing of these by just adding layers onto the end of the URL for the spatial portal, and you will get some metadata and information about all 320 layers that we currently have. An example would be precipitation in the spring, and that is the distribution of that characteristic. So that's an example of an environmental layer. In this case, you can see there are a series of polygons which are referring to a study area which may be able to limit analysis to that particular area. So it's an example of a contextual layer. So you can see there's marine layers and terrestrial, and in fact there are some world layers as well. So let's just map out a few layers. So what we do here is say add to map layers, and what we might do is just do a search. And again, if you know some of the shortcuts, distance to permanent water weighted, you can see here that I've developed a traffic light system that shows you the relationship between the selected layer and all remaining layers, where green says unrelated, yellow to orange says there's a relationship, and red says that there's a close relationship. That makes a lot of sense when you're doing some of the analysis, because you do not want too much correlation involved in, say, a prediction. So we'll just simply map that layer, and there it is. Like all the rest of it in the atlas, we can zoom to the extent of that layer, and we can, in this case, just change the degree of opacity so we can be able to see through it, or not, as the case may be. Let's just take one more. Let's have a look at salinity. This is a world layer, but you can see the salinity in the Australian region. So there's an example of two layers. What we might do is just switch off some of these to hide the complications. And what we might do now is look at what I would call a facet. So what do we want to apply the facet to? Again, there's this definition of area right throughout the spatial portal. What facet do you want? Well, we have quite a lot to choose from, as you can see. And the one that I'm looking for here is lifeform. As soon as I click on lifeform, you will see what the atlas has in the way of broad breakdown of taxonomic groups. In this particular case, I'm going to select fungi. This is the number of records associated with that in the whole of the atlas. In this case, 168,000 roughly. What it's going to do is it's going to map that facet. So in fact, there's all the records of fungi in the Australian region. So what we can do now is just do what we've done with anything else. We could actually say facet the facet on the collection type. So there is the coloration of the fungi for Australia, broken down by the collection that the fungi came from. As you can see, the greatest number of records have come from the Australian National Herbarium, but various other states and territory are included. We can again subset that on the basis of, in this case, the collection to create a new layer if we so wish. Let's just now have a look at some of the tools which use the data in the atlas. You've seen area report. I won't deal with nearest locality. You can click on a map and get the 5 closest points, and direction and bearing and distance. Points to grid, I'll just mention it here. We won't have time to demonstrate it, but you can create a species occurrence layer from an assemblage, if you like. In other words, eucalypts or acacia and acacia put together. You can then do a sample over all those occurrences and turn it into a science by species matrix if you want to, and on the fly also create a species richness layer and an occurrence density layer in addition to that. There's a whole series of criteria that can be used in that one, but we won't have time to do it. Let's have a look at the scatter plot. Incredibly simple, but incredibly powerful. So we'll limit the area to the world. We will, in fact, add a species in here. In this case, it's going to be one of the sandalwoods. .95 records in this case. We won't go for a highlight. We will just take the really simple, a couple of really simple environmental variables. They can be any environmental variables, by the way. So in this case, we're just going to take mean annual temperature and annual precipitation. You'll see what this option here does in a second. For a background species, we can use any of the stuff that we currently have, but we're going to add, in fact, another sandalwood. In this case, Leptomeria squirrelosa, 266 records. Thank you. Call it my scatter plot by default. And what you will see here is the scatter plot of temperature, precipitation for the environment that's contained by those particular species. The blue dots here, I'll just increase the size on the plot to give you a bit better idea. Might reduce the map ones a fraction. Now what this is the background grayscale that you can see is in fact the area of the associated combination of environments in Australia. In other words, we're only looking at a subset of the area here that's covered by the distribution of these two species, where the light colours represent large areas, the black colours represent very small areas. The large grey circles represent the background species, in this case Leptomeria squirrelosa, I think it is, squirrelosa, and the blue dots obviously represent the foreground species. Like everything else, we can in fact do a facet on the basis, in this case, let's just take Biogeographic Regions. So Australia is classified into bioregions, and what we're doing here is simply saying colour the dots up now on the map for, in this case, the two species on the basis of their bioregions, and you can see them not only on the map here but also the bioregions coloured up in the environmental space. If we want to find out where that particular occurrence record is, we can simply click on it, and there it is here. So we're matching the environmental space to the geographic space, and we can go back the other way, except we don't have time to demonstrate it all. What I'm going to do now is to just show you how one of the other techniques called classification. What I might do in this particular case is just pick the island of Tasmania, and what we're going to do is not use, in this case, any biological data. We'll this time use the area called current extent, which is the map window. There are a couple of predefined combinations of environmental data. I've actually identified 5 moderately independent terrestrial environmental layers. You can see the colour scheme that remains. So we have 5 variables here, which I think tend to explain quite a lot of the environmental variation in Australia. We can select how many groups we want, and we'll call it my classification. Here are the layers. We can export and import those if we want to, but seeing that they're 5 predefined, that's not usually a problem. In other words, we can always just go back and select that 5 set. So what's happening now is we're attempting to combine all 5 layers into one new layer that represents most of the variation that we see in those 5 layers. So it's attempting to provide effectively an environmental classification, in this case for the state of Tasmania, or the island of Tasmania. Why is that useful for such a biodiversity oriented tool? Well, the simple answer to that is that we do not have systematic environmental information for all of Australia, and